Hi everyone, my name's Sarah. Today I'm going to give a brief introduction to the latest EPI2ME platform and then show you how you can streamline your analysis using our sequencing workflows. Sequencing analysis can often be a bottleneck for research due to it being time-consuming, requiring a lot of compute resource, and not everyone always wanting to run things via command line. The goal of the EPI2ME team is to make analysis of nanopore sequencing data easy and accessible to everyone, and to free up researchers and organizations' time to focus on answering more important and interesting questions with sequencing technology. To do this, we've created considered and computationally efficient workflows, which you simply input your base called reads into and run, and they will output reports and standard bioinformatics format files that help you make sense of the results. To facilitate running the workflows, we've created the EPI2ME application, which is free to download via the EPI2ME or Oxford Nanopore website and available to run on Mac, Windows, or Unix. Once downloaded, you can run the analysis via the app, either locally on your computer, and now additionally also in the cloud. Here you can see a short demo of the app, showing how simple and intuitive the interface is. Each workflow comes with documentation explaining what the workflow can be used for, as well as an overview of the tools used, and a short demo which you can use to check the workflow runs okay in your environment. Each workflow can be run either locally or in the cloud. The cloud can be a good option for users that don't have enough compute resource locally, or if you want to run more than one analysis at a time. The workflows have minimal input requirements, with the main ones being your base called reads, and for this you can simply point it at the minnow output folder. There's also other advanced parameters with help text explaining what's required for each one, but most of these should be set to sensible defaults. We can then run a workflow and watch it progress, or we can navigate away and run more than one analysis at a time. Once complete, each workflow outputs a rich HTML report with interactive plots and stats specific for that analysis type, as well as standard bioinformatics format files such as BAMs and VCFs. And we've now also embedded IGV into the app, allowing you to visualize alignments without needing to launch an additional program. We're also working with third-party tertiary analysis providers, such as Day Zero Diagnostics, allowing you to launch some of the workflow outputs in their downstream specialized analysis and database tools. We've also created the EPI2ME file format, which allows users to confidently use and share workflows entirely offline, which is useful for users working in restricted environments. This is a compressed file type, which contains all of the code, references, databases, and containers required to run a workflow offline. You can find the code for all of the workflows on GitHub, where you'll also find the command to get you started if you prefer to run things via command line. You'll notice that all of the workflows are developed using the Nextflow Workflow Manager. We use this to help manage computational resources and ensure all our developers are on the same page. We also support containers, either Singularity or Docker, and this ensures reproducibility of results, and the containers contain all of the code and latest versions of the workflow needs to run. You're also welcome to develop your own pipelines using our template workflow. The benefit of this is you can then import the workflow into the app and make full use of the graphical user interface without any additional setup steps required. We now have 17 workflows covering a range of popular bioinformatics use cases. These represent best practice for analyzing nanopore sequencing data and have been developed alongside internal benchmarking and tool development efforts, and we also work with external tool providers as well. Many of the workflows are supported by end-to-end protocols, taking you from sample prep through to sequencing and analysis. You can find these protocols linked in the documentation for each workflow and also on the Oxford Nanopore website. I don't have time to go through all of the workflows in detail, but I thought I would just mention a few that I think is useful to be aware of, as they're quite broadly applicable and might be useful for your projects. Starting with the metagenomics or 16S workflows, these can be used for classifying what's in your sample and getting relative abundances, and it will output some nice interactive Sankey plots and sunburst plots, allowing you to explore the lineages of what's in your sample. Then we have the human variation workflow, which can be used for looking at germline variants, including small nucleotide variants, structural variants, copy number variation, and short tandem repeats. This workflow outputs a nice variant report for each variant type, and will also do phasing, so it will output some haplotag BAMs. Then we have a couple of workflows which can be used for exploring full-length transcripts. starting with the single-cell workflow, which can be used with our cDNA reads to look at full-length transcripts at the single-cell level. We've recently also added SNP calling to this workflow, as well as the ability to detect gene fusions. Then I also wanted to mention the transcriptomes workflow, as this can be used with either our cDNA or direct RNA reads, and this can be used for creating a reference-guided transcript assembly, which allows you to look for potential novel transcripts and also do differential gene expression analysis and differential transcript usage analysis. As well as maintaining and improving the existing workflows, we're also always releasing new ones. So new for 2025, we have the TeloSeq workflow. So for anyone that came to London Calling last year, you may have seen Carol Greider's talk on telomeres, so you'll know how important and interesting they are. The TeloSeq workflow allows you to get accurate measurements of your telomere lengths and also assign them to the various chromosomal arms. Then we have the TRIO workflow, which can be used for joint variant calling and pedigree phasing, so looking at an individual and comparing it with the biological parents, and this allows you to more confidently look into de novo variants. Last but not least, we have the pharmacogenomics or PGX workflow. This can be used for detecting genetic variants, predicting drug response phenotypes, and annotation of pharmacogenomic markers. If you're interested in this workflow, then please let us know to gain early access. So I would highly encourage you to go away and take a look at our library of workflows. I think it's really useful to be aware of what's available, as it might be useful for either current or future projects. Also, the workflows can serve as an educational tool to learn about the different types of analysis available and can perhaps inspire some broader use cases and projects in the knowledge that the analysis is going to be that much bit easier and quicker. Also, if you think anything's missing, you can let us know via the GitHub issues page or the Nanopore community. Finally, I wanted to mention the EPI2ME website, as it's a really great resource with lots of explanatory blog posts and how-to articles, as well as regular updates from the EPI2ME team. There you'll also be able to find some more comprehensive datasets that are part of the ONT Open Dataset project. These can be used for lots of things, such as developing your own tools, exploring characteristics of nanopore sequencing data, reproducing some of our benchmarks or looking into our benchmarks, and also developing your own tools. We have over 20 available, with some recent examples that we've published, including a genome in a bottle HG0 1-7 dataset, base called with a recent base caller, a plasmid validation set with some interesting repetitive region plasmids, and also a single cell dataset. Thank you.